The nervous system in organisms is very important because it helps animals detect external and internal signals, transmit and integrate information, and produce responses for any stimuli. This is a diagram of a nerve cell, also called a neuron. A neuron consists of the dendrites, which are branch-like fingers that receives information, a cell body, a nucleus, an axon where action potential takes place, and also where the message is transmitted through, myelin sheath, also known as swan cells, and myelin sheath helps facilitate and make action potential go faster, and axon terminals in the synapse, where the message is carried on to and passed to another neuron. Now I am going to explain how action potential is generated. At first, the axon is at resting potential, and that's when most gated sodium channels are closed. When the neuron is stimulated, which means when there's a message coming in, the gated sodium channels will open up and allow sodium to enter into the axon. And the inflow of sodium causes further depolarization, which will open up more sodium gated channels and will allow more sodium to come into the axon. Right before the final phase of action potential, which is called undershoot, the potassium channels will open up, allowing potassium to flow out of the axon. And this rapid outflow of potassium will bring the membrane potential back towards the falling phase. During the phase of undershoot, the membrane's permeability to potassium is higher than at resting potential. So basically the membrane potential is closer to falling phase than it is to resting potential. And eventually the gated potassium and sodium channels will close, and that will result in the membrane potential to return to resting potential, which is the very beginning before the stimulus happened. As for how action potential is powered, the sodium potassium pump uses ATP to actively transport sodium out of the cell and the potassium into the cell. Sodium is concentrated across the membrane, but very little net diffusion of sodium occurs because of the limited passageways provided by the sodium channels, as very few are open. However, the large contrast of open potassium channels allow a huge significant amount of outflow of potassium. This outflow of potassium results in a net negative charge inside the cell due to the weakly permeable membrane. This conducts action potential. Now let's see the synapse in action. For example, dopamine. The dopamine will travel from the axon to the axon terminal, and these dopamine neurotransmitters will release the dopamine out through the dopamine transporters into the synaptic gap. From there, the dopamine will travel from the synaptic gap into these dopamine receptors, which will allow the dopamine to transmit into another neuron through the dendrites. Dopamine affects the brain processes that control movement, emotional response, and also ability to experience pleasure and pain. Another example of the synapse in action is serotonin. After the serotonin neurotransmitters are done doing its job, the serotonin transporter's job is to remove the serotonin neurotransmitters from the synaptic gap. However, also, like dopamine, serotonin can be released from the axon terminals into the serotonin receptors generated by the stimulus. Serotonin is involved and plays a key role in regulating sleep, memory, learning, temperature, appetite, mood, behavior, muscle contraction, depression, cardiovascular function, endocrine regulation, regulating aging, bone metabolism, and wound healing. So our human brain consists of different regions that has their exclusive functions. Our occipital lobe is responsible for our vision. Our temporal lobe is responsible for our hearing. Our frontal lobe is responsible for reasoning, planning, parts of speech, movement, emotions, and problem solving. The parietal lobe is responsible for perception of stimuli such as touch, pressure, temperature, and pain. The cerebrum of our brain controls voluntary movements in our body, and the cerebellum in the brain controls and balances muscle movements. Our brainstem is responsible for involuntary muscle movements, and neurotransmitters work in controlling movements. Our brain is split into the left hemisphere and also the right hemisphere. Our left hemisphere controls what the right side of our body does, and our right hemisphere controls what our left side of the body does. A more deeper look at our brain is that the diencephalon includes thalamus, hypothalamus, pituitary gland, and pineal gland. Our brainstem includes midbrain, pons, medulla, and our cerebellum is located at the bottom, low backside of our head. The thalamus is responsible and is the main input center for sensory information going to the cerebrum. The cerebrum is basically the whole pink part of this diagram. The hypothalamus is where the body temperature is regulated. It is the central biological clock, and it controls the pituitary gland. It regulates our hunger, thirst, and plays a role in our sexual and mating behaviors, and controls our response to fight or flight, and it also regulates the source of release of hormones in the pituitary gland. The pineal gland is the source of melatonin, and it also contains one of several clusters of capillaries that generate cerebral spinal fluid from blood. Moving on to our brainstem, the midbrain receives and integrates several types of sensory information and sends it to a specific region of the forebrain. Also, the midbrain coordinates visual reflexes such as our peripheral vision reflex. Our pons and medulla sort of work together to effectively carry out their job, and their main function is to transfer the information between the peripheral nervous system and the midbrain and forebrain. Pons and medulla help coordinate body movements such as running and climbing. The medulla is mainly responsible for controlling automatic and homeostatic functions like our breathing, heart and blood vessel activity, swallowing, vomiting, and digestions. Basically, any activity that we would subconsciously do in our daily lives. And the pons help us regulate breathing centers in the medulla.